Okay, for chapter 2, tools of the laboratory, we're going to be actually looking at what it takes to actually study microbes, specifically in a laboratory setting. Now, as we approach this, we want to think of microbiology in the laboratory setting. We want to approach this with the idea of the 5 I's. Okay? The 5 I's being inoculation, incubation, isolation, inspection, and identification. And what we're going to do is we're going to go through each of these individually in this chapter and discuss in generalized terms what that means, what it takes. When you look at inoculation, inoculation basically means to culture, the growing of microbes. Here, growing is not like how you grew, how you went from a little kid to a big adult. No, growing in the microbiology lab setting means increasing in cell numbers. You start with a very few bacteria, and then 24 hours later you have billions. You're going to be growing these in what is referred to as a medium, an agar or a broth that supplies water and nutrients. Agar, as we're going to see, kind of a thickened water. Think jello. But this is made from agar, which originally was a seaweed extract. We're going to supply all the water a living cell will need and any of the nutrients. The inoculation here is taking a microbe and introducing it to an agar or a broth so that this microbe can grow, increase in numbers. Now, it's important to be able to do this because you don't always know what the source of your microbes are going to be. It could be body fluids, blood, urine, sputum, cerebral spinal fluid, whatever. Some anatomical swab. You've probably had this done at some point in time in your life. Throats, nose, ears, genitals. Maybe they scraped or took a piece out of some diseased tissue. Somebody has gas gangrene. You want to test them for C. diff. Or maybe it's not a medical thing. Maybe it's just an environmental. You want to know what bacteria are in the soil here. Once you've inoculated, you've taken the microbes, the bacteria, the whatever, you've introduced it to the media, the agar or the broth, you now have to incubate. It's going to take time. Because when you inoculate, you're only transferring over, you're only adding in a few bacteria, a couple of dozen, maybe a few hundred at most. And given time, these microbes will increase in numbers. 1 becomes 2, 4, 8, 16. You do that enough, and then within 24 hours, you're going to have millions, if not billions, of bacteria growing on the plate in the liquid broth. Now, this incubation, not only is it time, but it's also maintaining an environment. Temperature, turns out, is very important. The majority of microbes that will infect humans like to grow around 37 degrees Celsius, give or take a few degrees, 35 to 39 tops. So you'll put them in a chamber, a special chamber called an incubator. That incubator will hold the temperature of the interior that at whatever you set it at, 35, 37, whatever. Microbes have the water, they have the nutrients, they're kept at the correct temperature they need to grow. And this is important, being able to, inoculate and incubate. You look here, you can have what is referred to as pure culture, you're taking from a broth or a plate that has only one type of bacteria growing on it, and you're putting it into a new plate, a new tube, a new agar. Sometimes you have mixed cultures. You see here on this, in this tube, only the yellow bacteria here. Here is yellow. Over here is white. When you look at it on the agar plate, lots of little yellow dots, lots of little white dots. We're also going to want to grow things on plates or on agars so you can test for contamination. Notice here on this, it's all red, and on the plate, the majority red dots and just a few of the little white ones here. These white ones are something new, something you don't want to be there, hence they call it contamination. We're going to see that agars and broths are going to be classified, split into groups based on three criteria. First, what's the physical state? Is it a liquid? Is it a solid? Is it a semi-solid? If it's a solid, it means it's going to be firm. Think, really firm jello. Semi-solid, it's going to be kind of liquidy. If it's solid, well, is it supposed to remain that way? As the bacteria grow, increase in numbers, are they going to digest it away? Are they going to break it down? Yes, no. And then there's the chemical composition. Do we know everything that's in there? Down to how many moles of salt, how many moles of certain nutrients? Or is it complex? It's a little bit of this, a little bit of that added together, and it's a rich soup. And then the functional type. What's the purpose? General purpose, as we're going to see, is just for growing any microbe. Enriched, well, turns out certain ones are very, very finicky, selective, differential, anaerobic, and one's just for enumeration. Purpose built agars and broths. That's what these functional types are. So when you look at liquid to the left, it's just water. It's water with certain nutrients, certain dyes, certain pH markers. Turns out we like colors in microbiology. We're going to add in a certain dye, a certain compound that starts one color, and as the bacteria grow and metabolize something, it changes colors. Goes from red to yellow, green to blue. It's easy to tell these differences. Semi-solid ones, as you see in the middle there, are going to allow for bacteria to grow in and on and swim through. They're not free-floating like the liquid, but they're able to move around. And then there's the solid. Okay, like I said, has the texture of really firm jello. The bacteria grow on the surface, not in it. As I mentioned, the defined media, also known as synthetic in some textbooks. All components are known. We know down to the molecule, to the molar concentration, how much of everything is there. This is good for research. You want to test the limits of a bacteria to be able to grow. Well, you're going to limit certain vitamins, you're going to limit certain amino acids, certain salts, things like that to see, can it metabolize still? And then there's the complex. You loosely know what's there. It's going to have blood added to it. You don't know exactly what's in blood, how much, because from that sheep, that horse, that person. Or it's going to be tissue digest. You're going to take pancreas and digest it up. Brain, heart, muscle, boil it up. You know there's going to be fats. You know there's going to be lipids. You know there's going to be amino acids and proteins and sugars. You just don't know how much. You look at this defined media for the growing of Staphylococcus aureus. You know exactly down to the gram, and you can calculate the molarity of all the amino acids, all the vitamins, all the salts. And yeah, if you're going to make this, you do have to measure out 1.25 grams of magnesium sulfate per liter, 1.125 grams of iron chloride, half a gram of arginine, glycine, leucine. You have to measure these out. That's what defined media is. You know exactly what's there. Whereas on the flip side, complex, don't know. 27.5 grams of brain heart extract, peptone extract. Peptone, if you think back from your high school biology, peptone is an enzyme. I can't remember if it's stomach, small intestine, that helps digest, break down your food. So you're going to incubate it with the brain, heart. You're going to dry out brain. You're going to dry out hearts from sheep. You're going to grind them up, pat them in water, add the peptone. Peptone's going to break up. So again, we don't know how much and of what's there. We just know there's going to be lipids. There's going to be amino acids. There's going to be some sugars. How much? Don't know. But this is a soup. When you look at this is literally a soup, a broth, an auger that is so nutrient rich, this brain heart infusion broth, that you could live on it. You as a human being could actually eat this as your dietary source of calories.